And hey there, molecular genetics friends. So, we're going to just keep on rolling because, the semester's finishing up. I've made a mess of things, and what the hell? We're going to just do what we're going to do. Fear not. We'll have some stuff to talk about for finals, and I'll get things graded, and, we're all making A's. Let's just face it. So, a couple of thoughts. We were talking about antibodies and the creation of antibodies and how the B cell undergoes what's called somatic recombination, in that you build a light chain and a heavy chain gene. And there's just a vocabulary word that is useful. If one of the recombination events doesn't work, then the other chromosome can undergo recombination. But if the first recombination does work and produces a functional gene, which it usually does, the other one, the other chromosome, is inhibited. And that is known as allelic exclusion. And what this means is a B cell, once it undergoes somatic recombination, which is doing actually during embryogenesis, is producing a single type of antibody, a heavy chain and a light chain with the variable region. And then only if it binds antigen does it begin to go in, start dividing a lot. And then as it begins to divide, it'll start to class switch, and you'll go from having the membrane-bound antibody to forming these different types, like the immunoglobulin G, which is the circulating antibody. And so that's what vaccines are doing. They're essentially triggering a B cell, or a collection of B cells, to begin undergoing cell division and to start repopulating your B cell population. And within a few weeks, you begin producing circulating antibody that will hopefully neutralize the virus. Now, one of the tricks you can do, and this is sort of Nobel Prize winning stuff, back in the 70s, is you can take a B cell and fuse it with an immortalized cell line, tissue culture cell, and actually make a tissue culture cell line of that single B cell. And when you have an antibody or an antigen reaction, you're going to have multiple B cells can bind the antigen. Some are binding at the same spot, some are binding at different spots. The spots they bind are called epitopes. And different antibodies will be more effective at doing different things. But what you can do, and this is referred to as a polyclonal antibody because it's being produced by a whole range of different B cells, and it's a whole set of different antibodies. But you can select for a single B cell, and you can make what is known as a monoclonal antibody cell line. And this is a pretty big deal because now what you've got is, in fact, a tissue culture cell that is part just generic tissue culture cell and part B cell, and it's producing the antibody. And if you select the appropriate B cell at the appropriate time, you can have a B cell that was secreting immunoglobulin G into the circulating material, and you can now make a monoclonal antibody cell line that'll begin secreting the antibody into the cell media. And so this has been a pretty cool tool for cell biology, also a therapeutic tool. So what people do is generate monoclonal antibodies against all kinds of things. You can generate a monoclonal antibody against a gene or a protein that's involved in cancer production. So the various sorts of oncogenes. There are monoclonal antibodies that will target them. And the advantage is you do an injection to a mouse. You wait a couple of weeks, you take his little spleen out, you wash out all the cells, the B cells can continue to divide, you actually fuse them with a tissue culture cell line, and this is really where 96 well plates were invented, is so you do what's called limiting dilution, where you have basically one cell in each well, and you look to see if they're producing antibodies against the protein that you want. Once you identify them, you start to grow those cells out, and you have really for all intents and purposes, an immortalized cell line that is producing your antibody. So that's a monoclonal antibody. Very cool story. We can talk more about it if you're interested. Give me a holler. I thought what we would now turn our attention to is some basics of transcription, because really the to me, one of the coolest things about molecular genetics is control of gene expression. And that's what we're going to spend the rest of our time on. And I promise we're going to be doing a little bit more discussion about this. All right. So I will say that as far as developing some kind of in vitro hands-on lab, I've been a complete failure. So there you go. But we're going to continue on this. So let's think about transcription. Actually, we're going to start first with prokaryotic transcription. And first, I see my coffee's ready. I'll be right back. And those of you who know me, that you know that I can't be an effective lecturer unless I have my coffee. But prokaryotic transcription. What we're going to do now is we're going to take a portion of the DNA and actually have the structural gene being expressed. And so we denote the first base of the RNA as +1. And that means that we've got the promoter area here. The promoter really is doing one thing. It is the binding site for RNA polymerase. It is typically got an AT-rich region, what we know as the Tata box. This is a spot that is going to have a weaker interaction between the two strands, and so it can be pulled apart more easily to allow the RNA polymerase to get in, because what we're doing is we are using the bottom strand of the double-stranded DNA. The bottom strand is what we would think of as the template strand. That's what we're going to be using in reading and putting nucleotides in that will be the corresponding base pairs to generate the 5' to 3' top strand, but now in the RNA form. And really the only difference is instead of having thymines, we have uracils. So we've got the top strand running 5' to 3'. The bottom strand is running in the opposite orientation. All nucleotide polymerizations occur in the 5' to 3' direction, and that means that we're going to be going this way. Now, just as a point of standing on your head, there's no reason that you couldn't have a promoter over here running this direction and actually either overlapping or having portion of the RNA in this gene. So you might have the structural gene then would run like this for another gene. So having overlapping genes that run in opposite directions absolutely happens. Now, in advanced eukaryotes, we've got so much more DNA that we typically don't have overlapping genes. Bacteria sometimes do. Viruses always do. Viruses are extremely stingy with their DNA or their nucleic acids, and they'll have things running in both directions. And so it's just a matter of orientation. All right. The key point, though, is the RNA polymerase. So RNA polymerase is what's going to be binding to the actual DNA. And a key difference, and for those of you in molecular cell, you will hopefully remember this. The key difference is the prokaryotic RNA polymerase. It exhibits binding to DNA. It has an affinity for DNA. But the RNA polymerase exists in a couple of different forms. There's what's known as the core enzyme. And that is 2 alpha subunits, a beta subunit, and a beta prime subunit. This is the RNA polymerase that actually can carry out polymerization, but it doesn't have promoter-specific binding affinity. This exhibits what's known as loose or non-specific DNA binding. Now, what causes the RNA polymerase to bind specifically to promoter sequences is the addition of another protein, a fifth protein, known as the sigma factor. And this is a very cool kind of mechanism in that when RNA polymerase interacts with sigma factor, it now exhibits very tight binding to the promoter, and it can initiate transcription. Now, what we see is once transcription is initiated and you get about 50 nucleotides of the RNA in, the sigma factor can diffuse away. And so the core enzyme is able to carry out transcription, but we need sigma factor. And so, just as a little vocabulary word, the alpha 2 beta prime sigma, this is known as the holoenzyme, or the holoenzyme. And this exhibits very tight specific interactions with the promoter. Now, a particularly cool little trick is that bacteria will contain multiple sigma factors that will actually direct RNA polymerase to bind to different sets of promoters. Just as a little nerd point, the standard sigma factor is known as sigma 70. This is the sigma factor that allows for binding to the promoters to pretty much all the genes that a bacteria needs for standard operation. Now, the cool thing is, under conditions of stress or different developmental regulation, like some bacteria that can form spores, like bacilli, you have a sigma factor switch in which you'll have a different sigma factor, and that sigma factor will now direct the RNA polymerase to a different promoter sequence. And so you can have a class of genes that are only going to be expressed under very specific stress-like environments. And so the sigma factors, there's sigma 54, there's sigma S, sigma 32, I'm just reading this off the list here. These bind to different promoter sites, and some of them have really just one or two promoters that they can fire off of, one or two genes. And so things like dehydration, things like various sorts of environmental stresses, they can trigger a sigma factor shift, and you have a different set of genes that can now be transcribed. That's a pretty cool trick. And as we're going to start seeing when we compare this with eukaryotes, eukaryotes don't have a sigma factor at all. So that's something to keep in mind. Now, another thing to keep in mind as we're talking about transcription is this is all about transcription initiation. So we can control genes by deciding whether the RNA polymerase is going to bind to the promoter, transcription initiation. Now, another way we can control genes is a interesting kind of game that is really primarily prokaryotic, and it has to do with termination. And so let's just look at that for just a second. So, we've got we have the promoter sequence, and here's +1. Then we've got the end of the structural gene, and we're going to have some kind of termination sequence. Now, something to keep in mind, we're talking transcription, not translation. So when we start talking about this, let us remember, we're going to have what's known as the 5' untranslated region, and then a couple hundred nucleotides in is going to be the ATG, that is the initiating methionine, and this is the start of translation. And then we're going to have something like maybe a TAA, a stop codon. So we've got what's known as the open reading frame, or the ORF, and that is the region that is actually going to be translated, but transcription is going to have a longer stretch. So the RNA is going to be longer than the actual region that is required for translating a protein. And termination is an interesting kind of game. So termination is not going to be right next to the stop codon. Termination is going to be a couple hundred nucleotides downstream of the stop codon. And as we'll see, in eukaryotes, termination isn't nearly as big a deal. In prokaryotes, it's actually a pretty useful thing. And here's why. Or let's just think about it this way. The DNA sequences that are required for termination are located upstream of the terminator sequence. So there is a consensus sequence for termination. And what's happening is we form a hairpin loop. And you know something? We're going to go to some figures just because they're going to do a nice job of diagramming this. This is chapter 19 in one of the older books. And I, of course, will post the PowerPoints. So I'll give you these PowerPoints here as well. All right, so the sequences required for termination are actually being transcribed. And what folks have done is compare this sequence in all these different terminal ends and develop consensus sequences. And a couple of interesting things have come about. First off, very often the RNA is able to form a hairpin. That is, there's going to be complementary sequence that is going to have a stretch of non-interesting sequence that will allow for the formation of this hairpin. And this hairpin is essentially goosing the RNA polymerase and causing it to fall off. So now, this is known as an intrinsic termination. It is a sequence that's in the DNA forming this hairpin, and it causes the hairpin causes the RNA polymerase to fall off. Now, there's actually a fairly good consensus sequence, and we've got this palindromic region that's able to hybridize and to form a hairpin. All right, now the alternative to intrinsic termination is something called row-dependent termination. And this is where I wish I had my board. I could write on the smart board. But row-dependent termination, there's no pen. All right. Anyway, row-dependent termination is using a DNA sequence that is known as the row utilization site, or the RUT site. The row factor binds to the RNA that is transcribed from the RUT site. It then begins to move up the RNA. The polymerase is going to pause at a hairpin, and row is going to actually interact to unwind the RNA-DNA hybrid and release the RNA. And so this is a different kind of termination sequence. And the way you know it is, you look for the RUT site. If you've got a RUT site, then that means this RNA is being terminated by row-dependent termination. So here's a RUT site, just for your own information. It's got a fairly high C content, poor in G, and this is at the 3' end of the RNA. So row is actually able to interact here. Anything here? Sigma factors can be substituted, as we mentioned. Here's just a list of things that sigma factors can do. So sigma factor 70 is the standard. Sigma factor S is some stress responses. Sigma factor 32 actually is part of heat shock. Heat shock in bacteria will cause them to shut down transcription of everything but a set of heat shock proteins, and they do this by having a sigma factor shift. Yeah. All right. Now, another clever trick of eukaryotic RNA polymerase or prokaryotic RNA polymerase has to do with termination, in that there is something called anti-termination. We actually can inhibit a terminator and allow for read-through. Now, let's just think about why this is important. If you recall, eukaryotes, when they are translating an RNA, they bind the 5' end of the RNA molecule, the cap, and they start working their way through. When they find, when the ribosome finds the ATG, it begins synthesizing a protein. When it hits the stop codon, it falls off. Now, that's all it can do. So you can only translate the first open reading frame of an RNA. And for most eukaryotic RNAs, there is only one. Now, with prokaryotes, they can be what we call polycistronic, in which you have an RNA that can code for multiple separate proteins. And this works because the RNA polymerase can not bind to the 5' end, not the RNA polymerase, excuse me, the ribosome doesn't bind to the 5' end. The ribosome actually binds at the ribosome binding site. So the ribosome binding site is kind of like a landing pad, sort of like the helicopter model, where it just lands in. What this means is you can have an RNA that might contain more than one open reading frame, and this second open reading frame, now it actually can be translated. So anti-termination is a very clever way of extending an RNA out, so now you might code for an RNA that'll have the ability to generate more than one protein. Clever, yes? So you've got the promoter, and in the case of standard termination, the RNA polymerase is going to make its way through to the first terminator. It stops. It falls off. Anti-termination, though, allows for read-through. The RNA polymerase starts moving through. It hits the terminator, but because there is an anti-termination factor, it moves on through, and it generates a longer RNA that most likely is going to have another gene, another open reading frame. And so this is a way we can actually control the polycistronic nature of an RNA. And it's actually a very cool little trick. So there are a couple of different anti-termination systems. One of the best defined anti-termination systems is an anti-terminator that is known as N. Why the creative name N? I don't know. But N, let's see, anything there here? N is using a site, the anti-termination site, that is known as an N utilization site or a NUT site. N can bind and allow for the RNA polymerase to read through. It allows for the RNA polymerase to pass through a terminator spot. And so this allows for the production of a longer RNA. And we're going to see, we start talking about a little bit of interesting viral gene regulation. And N anti-terminator is going to be playing a critical role in bacteriophage. So that's just another thing to keep in mind. Lastly, in bacteria, and just another difference between prokaryotes and eukaryotes, the messenger RNA is being transcribed and then immediately being translated. And once it's been translated, that RNA is being degraded. It's all happening at the same time. What this means is that an RNA is being produced as soon as the ribosome binding site is revealed. Ribosomes bind there and start translating, and the RNA polymerase is just staying just ahead of it, producing more RNA to be translated. And then we've got the protein coming out. The ribosomes hit the translation stop, and they can diffuse off, and the RNA polymerase hits its termination, and it stops. But as the RNA polymerase is making RNA, the 5' end is being chewed up. So RNA is notoriously unstable in bacteria. It doesn't have a very long half-life. And we'll see that in eukaryotes, the half-life of an RNA actually is a pretty cool developmental control mechanism. Depending on what your gender is, females have RNA present in the eggs and the oocytes that was produced basically neonatally, and you've been holding on to it all of those years. That RNA is quite stable. Gentlemen don't typically have RNAs that are that long lived, but in oocytes we do. So bacteria, because of the way they're able to operate, they've got some clever gene regulation techniques that just aren't going to be found in the eukaryote. So this is a way we can look at the transcription unit in a bacterium. So this thin line is DNA, and what we've got here is the start of a transcript. The RNA is being produced, and as the RNA is being produced, the ribosomes bind and start making their way down. So the ribosomes are pretty big, and they actually are rather electron opaque. And we end up with an RNA that is actually quite long and completely covered in ribosomes. So, as we were saying, the bacteria have this untranslated region, and then if it's a polycistronic system, you have what's known as the intercistronic space. And it can range from a few nucleotides up to 40 or so. And then you have the next start codon of the next open reading frame. And so what this means is that you might have a terminator sequence here that you can be subject to anti-termination, and you can have an RNA that stops here, produces one protein, or an RNA that continues on and produces two proteins or multiple proteins. So this is some of the stuff that we're next going to be thinking about as we get into some of the ideas of gene regulation. And the idea here is to compare prokaryotic with eukaryotic. All right, I have been kind of on the short end of things for molecular genetics. I'm usually a train wreck, but not usually this big a train wreck. And we'll see where we go from here. All right, thanks guys. I will be talking with you later. Bye.